So now we do have our next speaker, Professor Dr. Wildon Aruni. He's a Pro Vice Chancellor of Satyabama University. Dr. Wilson has taken up the position of Pro Vice Chancellor of Satyabama Institute of Science and Technology, Chennai, on sabbatical leave from Professor Department of Microbiology, Infectious Disease and Global Health, California University of Science and Medicine, Loma Linda, USA. Principal Scientist, U.S. Department of Veterans Affairs, Loma Linda, CA, USA. Adjunct Professor, Stem Cell Core Facility, University of California, Riverside, USA. He has served in various academic and administrative positions, especially in the University Academic Affairs, Curriculum Revision, and Accreditation in the United States of America. He has been involved in active teaching and research for a period of 24 years. Previously, he was working as professor at the Center for Excellence in Animal Biotechnology and Immunology, and also at the Department of Microbiology, Madras Veterinary College, Tamil Nadu, and Animal Sciences University, Tanuvas, Chennai. During his tenure, he had occupied various academic capacities as head, University Bioinformatics Center, and Advanced Imaging Facility. As an experienced academician with a vast international exposure, he's also a well-trained researcher in the field of microbiology and biotechnology. His research focuses on studying infectious pathogens, their virulence, and their importance in systematic diseases. His expertise in the field of bioinformatics and nanovaccine delivery system is well acknowledged. He received his BVM degree from India, and his doctoral studies focused on studying the virulence of RB viruses and work performed in studying their molecular morphogenesis at the All India Institute of Medical Sciences, India. His postdoctoral training at the University of California, Davis, brought both seminal publications, that is USDA and BBSRC UK. His pioneering works also culminated in 2 patents. One of his other pioneering work through postdoctoral fellowship at the Laboratory of Gene Structure and Expression, National Cancer Center of Singapore, studied the haplotype variations on the enterocytes and the hepatic expression of P glycoproteins. He is also trained through an NIH postdoctoral research fellowship exploring the molecular aspects of human microbiome interactions. His research work at the Loma Linda University School of Medicine is on studying the virulence, determinants, and host pathogen interaction of oral pathogens and their causal role in systemic diseases through an integrated proteins approach. His studies were the first to explore a new oral pathogen, Filifactor allosis, and its virulence potential. His publications have been featured in the cover page of prestige journals like Nature Communications, Infection and Immunity, Proteomics, and in other peer-reviewed journals of high impact factors. He has been invited internationally to present his professional work and currently serving as adjunct faculty in many institutions abroad. In addition to his research initiatives, he has completed 4 extramural grants funded by DBT, DST, and ICMR, and currently funded with 2 grants from National Institute of Health, NIH, as a principal investigator. His research work has culminated into 88 publications, 3 books and 5 book chapters. He has a strong record of mentoring more than 21 graduate and 8 doctoral students in India and in the United States. His students have been well praised in reputed academic and research institutions internationally and nationally. He is currently serving on editorial boards of high impact peer-reviewed journals in the field of medical microbiology, biotechnology, and bioinformatics. He has serving as member of task forces in DBT and DST. He is also currently serving as a member of National Institute of Health Grant Review Study sections task forces, serving as a member of the Grant Review Panel on Vaccine Research Initiatives Grants, Bill and Melinda Gates Foundation, and a current Grant Review Panel member of Medical Research Council, MRC, and BBSRC UK. With this introduction, I would like to welcome Professor Wilson to give his keynote address. Welcome, sir.
Thank you. Thank you very much, Dr. Rajesh. Welcome. Can you just give me an access for sharing the screen, please?
Yes, sir. I made you as a presenter.
Okay. I hope all can see my presentation, right?
Yes, sir. It's visible, sir.
Okay. Thank you so much, organizers, for giving me an opportunity to give the keynote address in this August gathering. It was 415 years ago in 1600s, where when the first microscope was identified and it was invented. It was Antony von Leeuwenhoek who found little organisms which he named as animalcules. And he found that in a small piece of a cork. Later on, the development went on up until studying microbes everywhere, especially in every part of the environment. No matter what environment you would take, you would see microbes everywhere. There are microbes in the soil, there are microbes in the water, there are microbes in the sea, there are microbes everywhere, inclusive of in our body and outside our body too. So there is a lot of leap and bounds with regard to the research development, which you could see here, where people had moved from human genome project, where people started studying earlier the genes and the genome of human being. They now transitioned into studying human microbiome projects since 2008. And each and every microbiome plays a very vital role. So then what are microbiomes, by the way? They are a collection of the microbes which live in our body and outside our body, which clearly sets a very good environment interaction with the host. So the talk caption would be the human microbiome and the hidden health factors. So what if the research is not going to be useful for the human mankind? What if the research is going to just culminate in only publications and patent? What if the research is not going to be useful for the longevity and the health of the human being? Hence, this is a very important burning topic in the whole world where people are focused on studying human microbiome. There are two important captions in the whole world. People are just chasing the wind. Number one, people are trying to live healthy and people are trying to age healthy. These two are very important things which are being sabotaged by many environmental events, such as the ones that is currently going on, the COVID pandemic. So having said living healthy and aging healthy, people focused on three important factors with research and development going on, with the initial focus on the genetic part of the human being. People thought that the genes played a very vital role in the longevity and health, and it is only our parents who give the wealth to us. Later on, there was a different school of thought which said that the environment and the environment modifications play a very vital role. For example, a person who smokes will not live longer or will not live healthier because of the epigenetic changes that happen in the gene because of the environment. The recent trend is that environmental interactions play a very vital role. There are constant environmental niches which are there where we are already interacting with since the day we are born, such as the air that we breathe, the water that we drink, the organisms that live inside our body, etc. So I'm going to focus on the third important point, the environmental interactions. So having said initially the genetics was playing a very vital role in the longevity. So when you see this picture of the whole world, the color coding says that the more it is yellow or orange, the less the people lived in these places. The bluer they are, the people live longer. For example, in the United States, the average longevity rate is 82 years and above, whereas in case of humans in India, it is only around 65 to 68 years. So initially, people started chasing research, and I'm giving you two important examples here. Number one is the first known centurion who had lived for 100 years, documented in history, Jean Calment. Yes, she lived for 100 years. But whereas when you just see this lady, she is a chronic smoker. So, despite of her smoking habits, she lived for 100 years. And people said that genes are very strong, and she had a very good genes that made her live long. On the right side, you see a marathon runner named Jim Fix, who started running marathon only getting very scared after his father's death at the age of 45. He thought that he will also die very early. So he started running at the age of 40. When he started running at the age of 40 marathons, he was found dead running a marathon at the age of 42. So he couldn't beat the genes. So on the left side, you see a person who beat the genes. The other one couldn't beat the inherent property that we inherit from our parents. Having said this, the life expectancy in the Indian population, what you see here on the left graph, is around 66 to 68, whereas compared to the all developed countries, which are very high. What really clearly makes a difference in this health and longevity, especially in this part of the continent versus the other places? The initial research on health and aging dated back to 1935, when a scientist called Mackay said that if you are going to eat little, you are going to be very healthy. So you see two monkeys, one on the left side, another one on the right side. On the left side, this monkey was given a restricted feeding versus the right side monkey, which was given ad libitum feeding. So the more the monkey ate, more the merrier it was. But at the age developed, it became so sober and it became so sluggish. This was the first experiment by Mackay. He wanted to prove this, so he himself went into a diet restriction to see how health and longevity played a very vital role. On the left side, you saw the same person, the author of this one, with the high body weight, okay, the vitals, the cholesterol, fasting glucose, blood pressure, all very high, versus on the right side, where it's a controlled eating habits which made him so healthy. Even you could see this in my life, where when I was in India some 15 years ago, you would see here I was so 50, and whereas this is what I look now, with an age difference between 35 to 55. So is this really making a very vital, important role, as the food plays a very vital role in health? Where people went to 5 different places in the whole world where people lived more than 100 years. These places or these spots in the whole world are called as blue zones. One among the blue zone is the place where I come from, Loma Linda University, Loma Linda, California. So people thought that there should be something different within these people here, the habitat, which played a very vital role that increased the longevity and good health conditions versus the other geographical area. There were a lot of books that have been written on diet, life expectancy, secret of living long, etc. in the blue zone. And what you see are our publications from the Loma Linda University. And the Loma Linda legacy had started since very long, where you might know that the Kellogg corn flakes was first invented in Loma Linda, and these two are the Kellogg brothers, who gave their whole wealth to our university, where the university is sustaining on research by their own endowments. So with this legacy of good habits of feeding, a long study on longevity was conducted by National Institutes of Health in the US. It started in 1955. It is still going on. And this study is not through any lab animals, but with the real human being, where they are taking into consideration different feeding patterns or food patterns by which they eat, what is the size of the meal that they eat, what is their physical activity, or what are the parameters that play a very vital role from the environmental, etc. So what you see down is a a total swimming pool exclusively for people who are 70 and above. And this is the food. And I should say that in Loma Linda, everybody is a vegetarian, and there is no smoking within the city limits. So they say that being vegetarian makes you live longer because of certain obvious reasons, where I'm going to just discuss now. But whereas when you just take our Indian counterpart, we live to eat and we eat to live. And people are very much fascinated about eating all means of food, and this includes me as well. And people who ate restricted diet with the vegetarian base really lived longer. And what you see, these two young people here are our administrative peoples of the university who still live and are very hale and healthy. One important thing is the activity that played a very vital role, where people there at Loma Linda, they run marathons very well. As I was just telling you, an example of gym fix. So what you see here is an 85-year-old, our Indian American, who's a cardiologist, who is our marathon guru. And what you see here is me, who's a beginner long before in a marathon. So why am I saying all these things? I'm talking about vegetarian diet. I'm talking about running marathons. They are all environmental cues that make us live healthier and longer. So coming back to the system again, genetics and genes had been proven false. Epigenetics is somewhat proven correct, but whereas they are not the real culprits for a health and a diseased state. The environmental interactions are the main focus that is given now by the research scientists. So we are going to talk about the microbiome, which is nothing but the environment a human being is surrounded by. And hence, they are called as human beings are called as superorganisms. So human beings are different because each and every human being are unique by their microbiome or the bacteria or the viruses that live within us. The microbiome totally, if you're going to take them, outnumbers the human cells by 10:1. If you have 10 cells of human cells, one human cells to 10 bacterial or microbial cells. 4 pounds of the total dry weight matter is only the microbes. With such an effect of microbes within your body, what you would see here is an analogy is that the 10 trillion human cells would account for 100 billion microbial cells. So to say that the red part which I'm just showing you here are only the human beings, whereas the white part that you see is all microbes. So now you imagine each and every one, just close your eyes and imagine that how many microbes live within us. That creates a very constant environment that is going to be interacting with us. This is being taken care initially by researchers, where people started taking the microbes from obese individuals and lean individuals. They gave it to lab animals to see how they look like. And they found that obese microbes that are taken from the obese individuals made the mice so big or obese, versus the lean microbial content taken from a lean person made the mouse very lean. So now people even had patented certain bacteria within these lean kind of stuff. One good example is that there is one bacteria that is being patented in Japan, where you might have seen Japanese who are always slim despite of eating more food. So the community assembly that takes from the mouth until the rectum where the feces goes out, bacteria and viruses and the microbes are there everywhere. And what you see here is the last column that I'm telling you is that the more you reach rectum, there are more cells that you see here. So these bacterial cells are constantly there in your system, and they play a very vital role in your health and in your disease state. So this is one of our good examples of recent publications that has come out from one of our collaborators, where he has taken a very extensive Western lifestyle, non-Western lifestyle from countries like Asia, and he wanted to see what is the real thing that is happening with all those people. And he found that more than 154.73 microbial genomes have been initially found, where 77% were very new. So to say that the microbes that live in your body are not even named now. There are a lot of microbes which are waiting to be named. No matter we are talking about E. coli, Salmonella, etc. But whereas you need to know that there are 77% of the microbes that dwell within your system are not being named, and they are very new, and people do not know what are the microbes. And they vary between the Western lifestyle and the non-Western lifestyle. It's just because that the food habits are entirely different between these two group of population. So what really happens when you take that kind of a food? There's a constant tussle between the health and the disease state. When if it is going to be a healthy or a normal state, there is symbiosis among the bacteria and the viruses that live within our body. But whereas it slowly progresses when there is going to be detrimental bacteria that enters inside your system, which makes a symbiosis a state called dysbiosis. Why this is more important? Because it is only the bacteria and the viruses that dwell within the body. They produce vitamins for you. They prevent you from the disease. They enhance the immunity. For example, in case of COVID conditions, etc. And they resist colonization of detrimental bacteria. So hence, this is more important that keeping a good microbiome within our system is very important for the healthy state. Why is that? Because these bacteria and viruses, they produce antibiosis, and they also produce probiosis, or the probiotics that produces good bacteria to be grown there. And ultimately, both together form the microbiosis that exists within our body, which is constantly fighting the bad bacteria. So the pictures which I'm showing you here is Alexander Fleming, and what you see here on the top is the Penicillium notatum that is producing penicillin. But whereas the one that you see in the blue color agar are the bacteria which are called as New Delhi beta-lactamase 1, which is a very detrimental bacteria where no antibiotic is available. So antibiotics are being used very scrupulously, but we do not know that the antibiosis happens within your body. But when you are going to take this kind of pill, you are going to just kill all the good bacteria within your system. Antibiotics are there everywhere, starting from antibacterial soap to deodorant to face cleansing products, toothpaste, mouthwashes, etc. Not alone the microbiome of the human being plays a very vital role. The positiveness of the microbiome of the soil I've shown you here. A very important species of bacteria, Streptomyces, produces a lot of good antibiotics that are currently in existence now. Not alone the soil microbes, but also what I'm showing you here is the gut microbiome of nematodes that is being used for producing the recent antibiotic, which is called as Darbact. So the human microbiome, the soil microbiome, the microbiome of even the worms, the microbiome of even the whole environment, they all collectively bring about the antibiosis of protection and immunity. Hence, antibiotics are now not being trying to be used, but whereas there are a lot of alternatives to antibiotics that have come here. One good important thing is the peptides and the small molecules that are being produced by the microbiome. So I'm just showing you here antimicrobial peptides and the modifications of antimicrobial peptides that are currently being produced by the microbiome, which are being used for clinical use. One good example is the lysines, which are being used to kill the bacteria. It's like an antibiotic where these lysins are being added with fortified with endolysins to produce artilysins, which are very important and potent lysins. They are being produced only by the bacteria that are present within our body. So ultimately what we get is a very good antibiosis that we get. So I'll show you two videos over here. Number one is the ordinary lysine here. You just see how long it takes to kill the bacteria in vitro. So it takes longer to kill the bacteria, and the bacteria is not killed properly. Whereas the RT lysine, you would see here, the bacteria is killed within a matter of quick seconds. So I just want you to remember that they are being produced by the microbiome. With such a lot of diversified microbiomes, which I'm showing you here, the fecal microbiome, the oral microbiome, which is here, skin microbiome, etc. What happens is this microbiome begets prophylaxis by competing with the detrimental pathogens or protecting the mucosal barrier, modulating the immune system, and also giving an antimicrobial peptide kind of a stuff in protecting the environment. So ultimately, what do we need for a good microbiome to exist? We need a good diet that will produce good microbiota, and ultimately the host is being protected. So a balanced diet with high fiber will bring balanced high diversity of anti-inflammatory microbiota, and ultimately a balanced healthy symbiotic life. The vice versa happens in an imbalanced non-symbiotic context, causing the disease. So this kind of diversification is the one that we see within our human system. So this is the map of diversity of the human microbiome that exists within our system, where you will see such a lot of diversification that exists within our body. How does this diversification happen within our body? Is just because of the food that we eat. Okay, if that is the case, is dietary diversity playing a very vital role? But whereas you need to know that only human beings eat 12 species of plants and 5 species of animals. Rest, we do not eat anything. And more than 150 to 200 species are only being used among the 250,000 to 300,000 edible plant species. So you can just think about how you eat and what you eat. There is no diversity in what we eat. For example, a human being, an Indian, in the morning he takes idli, afternoon rice, evening dosa or something. So there is no much of a diversity in the diet, but whereas diversity in diet plays a very vital role in good microbiome. Why? Because they produce a lot of molecules that bring about many variations and many phytostimulants, pigments, enzymes, etc. For example, this is a lettuce that I'm showing you here, which has a novel bioactive compounds. Okay, the greens that we are going to eat has certain bacteria called Enterobacter, Colitotrichum, Composis species, etc., which exist within as an intracellular state within the bacteria, within the greens. So these are all a couple of studies that we did it. As I was just talking about the 1915 study that happened through the progress. What I'm showing you on the left side are the fruits and vegetables that help in the longevity and health. So we have just done this with vegetarians, lacto-vegetarians, people who take only milk, pesco-vegetarians, people who are vegetarians but they take fish, semi-vegetarians, they take egg. You would see here pure vegetarians are the ones where they are going to be very good health, and these are the fruits and vegetables that play a very vital role or supplement in health and longevity. Starchy foods, okay, they are all negative fats. Fat, sweets, and snacks, which Indians are very, very known for. And they are also being corroborated with certain important cancers, for example, breast cancer, colon cancer, and prostate cancer. You see here the green block here represents the vegetarian, the vegans, where you would see here they have the less incidence of all the cancers, three collectively. The same way we did a multivariate hazard analysis between male and female data together. So what does this actually do? It takes into consideration the whole environment in which he or she lives, with the diet that he is going to take for quite a long time, and what is the assimilation pattern that takes care of modification of the microbiome. And ultimately, what is good and bad is being reported here, where the vegetarians produce the lowest multivariate hazard analysis. It means that the vegetarians are more protected when compared to all the non-vegetarians or lacto-vegetarian or pesco-vegetarian, semi-vegetarian, etc. Likewise, I'm just showing you the same for a prostate cancer for the males. Having said that, what is so unique with the plant diet versus animal diet? So we did a very extensive study with plant diet and animal diet, not with the guinea pigs, but whereas with the human beings, and we found certain important bacteria which are highly upregulated in the system. For example, they increase the short chain fatty acids, which reduces the inflammation. But whereas animal proteins increased TMOA, trimethylamine oxide, which causes cardiovascular disease. But whereas they reduce short chain fatty acids, and that is how they do not protect the individual. Likewise, the gut microbiome diversity is being studied, and we studied with 3 different diets. One is gluten-free diet, another one is Western diet, another one is Mediterranean diet, and I've shown you the data between the Western and non-Western diet, where Mediterranean diet comes from the Mediterranean region like Saudi Arabia, the Muslim countries, where Western diet from the West, and gluten-free diet to some extent from the Spanish side. And we found such a lot of variation in the gut microbiome. And what I've selected and showing you is the beneficial microbes that are there that bring about a very good positive note to the health and longevity. This is the data to back up what I have done earlier, what we have just given earlier, where this gives a species abundance of the good bacteria in different places of the microbiome. In the oral microbiome, in the mouth, in the gut microbiome, stomach, small intestines, etc. Inclusive of anterior nares. It means that in the nose. So bacteria do live in our air cavity, which can protect, play a very protective role. So this is how the relative abundance have been identified in a positive individual. For example, this is being done with the candidate subjects of lower letter per se. So how do they go about? Because they produce certain small molecules, and I've just given you on the right side certain small molecules that are being produced from detrimental bacteria, which are currently used positively for treatment. So what we did was we did a genomic approach versus metabolomic approach. See, altogether, when you just see and study the metabolites of small molecules, the metabolites of small molecules in the microbiomes are very rich. And you would see 1 into 10 to the power of 30 exists in the whole biosphere, in the whole environment, which is being not tapped, where approximately 25,000 is being well studied here. So what we did was we took this microbiome study. We did a very extensive study to see what is the interaction, what is the positive, negative interaction. I don't want to go very deep into the data, but what I'm showing you is we have taken, for example, PG is a bacteria called Parvovirus gingivalis. VP is Vallinella. Fusobacterium FN is Fusobacterium nucleatum, etc. So what we found was, with the base of the bioinformatic analysis and the proteomic approach, we found a lot of small molecule candidates that I'm just showing you here. And to make them sum up, we found a lot of small molecules that produced by certain important bacteria. And these small molecules are also increased in capacity, increased in concentration when people eat a lot of good diet such as ginger and certain important mushrooms called as shikate mushrooms. And these two are found to be high immune boosters of the diversified microbiome. So to say that it is only the diet that diversifies a very good microbiome. And this is a smoothie candidate very commonly being used in the Western world. So to make it a nutshell, when you do a research with the microbiome, the ones that you see in red are all very detrimental bacteria. The ones you see in blue are all good bacteria. But there are black dots which represent certain bacteria which neither become good or not become bad. But at the same time, when more good bacteria are there, this black becomes more blue. When more red bacteria are there, the black becomes more red. It's like a mischievous class where many people become so mischievous, etc. So microbiome played a very vital role. Why? It's just because if you take diets with vegetarian, you also take soil microbiomes as well. And ultimately, we did a very extensive analysis with two important bacteria that you will see here. And this is an electron micrograph that was published in the cover page of Infection Immunity, which said that when two bacteria join together, they produce hell a lot of havoc with the human cells. You could see here the whole human cells become so rumbled and stratified like a noodles, whereas the insect that you see here on the right side are a single bacterial candidate affecting the human cells. And you will see the variation in the human cells where the human cells are not corrugated. We also wanted to know what is the main reason. So we did a whole global proteome variation of the human host cells, and we found a lot of studies. One important finding was that certain group of bacteria called Odoribacter produce certain important small molecules which are very positively helpful for the microbiome to combat disease conditions. So many bacteria from our study we found to be used for reducing obesity, to reduce arthritis, to reduce peptic ulcers, to improve immunity, and certain important lactobacillus species we uniquely identified improved overall health. So now the question arises that if we say that Western world, the Eastern world, which group of people have the good microbiome? And this is a study from one of our collaborators from Harvard, where they have found out a group of people in Tanzania called Hasda tribe, where the people who had the very extensive, good, diversified microbiome. And I should say that from their feces, bacteria have been identified and they are being used as probiotic desiccated cultures. So this is the ones Hasda tribe hunters. And these are the diversified microbiome candidates that are being used for a diversified microbiome study. So now comes a tussle between biology and chemistry. As I've already told you, we were talking about microorganisms or the microbiome. And these microbiomes produce small molecules, which is nothing but the chemistry. So now, which is really good or which is really the best, biology or the chemistry? It is always a very big question between the biology and the chemistry. But whereas if you people could remember this particular thing, a Tamil this thing from Kalangar TV, Manada Mailada, where everybody says that your chemistry is important, chemistry is important. Ultimately, we are in a condition where the chemistry is not at all working anywhere and we are totally isolated within a single room. So now the chemistry is not working anywhere. But ultimately, we need to know that biology always wins here. The reason being the recent havoc of evolution of this pathogenic coronavirus that we are facing right now. It is just a biology, a slip of the biology mistake that has been made in a high security laboratory at Wuhan, which has brought a very monster coronavirus, COVID-19, that had got disseminated everywhere in the world, that is creating a lot of problems. And you will see here, and this is being ultimately published, where there are a lot of other things that happen too. Because when you just take the total human population into concern, there are only 3 places in the whole world where many high security pathogens are being protectively stored, such as smallpox, plague bacteria, etc. But whereas this is a recent thing which I'm just showing you here, is that there is a settling loss of a smallpox vial from a high security laboratory. So imagine biology always wins here because biology creates the chemistry. And I should certainly say that they become the nature's weaponized means where the biology is being weaponized to produce a monster super SARS virus that we are focusing on. As you all know, that the experiment to produce a super SARS virus had mishap to produce a COVID-19 mutation in bringing about the COVID-19 pandemic that we are facing. So having said that, with all Indians suing the Wuhan BSL laboratory in China, biology or chemistry, ultimately, this is the century of biotechnology. Where initially people started exploring biology. They were isolating new bacteria, and then finally they moved on to designing biology. It's like they started cloning genes. But now people have started building biology. It means that they are producing new entities of bacteria. For example, the one which is being published in Science 2019 by Craig Venter, you see on the right side, a pioneering man who had first synthesized 482 protein coding genes with 43 RNA genes to produce a pathogen, to produce an organism called Mycoplasma genitalica, which is the first synthetic microbe to be produced. So starting from exploring biology to designing biology through cloning, we have ended up in building biology. And people now are focusing on genetically engineered machines, which could do a very synthetic work. So I was very privileged to do my postdoc in J. Craig Venter's Institute. What you see on the right side is Craig Venter's Institute, and what you see here is me. And this is our small group at Loma Linda University who were very helpful in carrying a part of the research. I have not given very extensive data about what I presented, but I just wanted to tell it on a sequence to tell you that microbiome is a very potent environment that plays a very vital role because it has constant interaction in bringing good or bad to your system. So these are the couple of references from us. And to talk about Loma Linda University, couple of good things that we have. We were the first one to be patenting the cardiac stent that currently being used for heart attack. And this is the cardiac transplant. These babies are being transplanted with the new heart, and he is Leonard Bailey, doctor who did that. We have a proton therapy center, which is state of art in the United States. And I also represent Loma Linda University. What you see here on the right side is the Center for Research, Center for CDD, Center for Drug Discovery and Development. What you see here down, we have a biosafety level PSL3 laboratory. We do very extensive research on human pathogens. And this is a screenshot of our laboratory, and this is Satyabama. We are both academic as well as of a research component blended together. And with this small note, let me thank all the participants for giving me an opportunity to share my views about one of the important integers of environment in this environment day, which is nothing but a microbiome. Thank you very much.